Hello, my name is Hossein Ardohali from Northwestern University. I'm also the immediate past president of the American Society for Clinical Investigation, or ASCI. It's my pleasure to introduce two remarkable investigators who shared the 2022 Harrington Prize for Innovation in Medicine, Drs. Michelle Nuzenswag and Dr. James Crowe. As a reminder, the Harrington Prize was established in 2014 by the Harrington Discovery Institute in partnership with the American Society for Clinical Investigation. The Harrington Prize honors physician scientists who have moved science forward with achievements notable for innovation, creativity, and potential for clinical application. Dr. Nuzenswag earned his PhD from the Rockefeller University in 1981 in cellular immunology and received his MD from New York University School of Medicine in 1982. He completed his internship and residency in internal medicine and a clinical fellowship in infectious diseases at Mass General in 1985. From 1986 to 1989, he was a postdoctoral fellow in genetics at the Harvard Medical School and returned to Rockefeller University as an assistant professor in 1990. He's currently a senior physician and the Zanville A. Cohn and Ralph M. Steinman Professor at Rockefeller University. Dr. Nuzenswag's studies the molecular aspects of the immune system's adaptive and innate responses using a combination of biochemistry, molecular biology, and genetics. His laboratory has isolated and cloned human antibodies to HIV and other viruses and has explored their roles in prevention and therapy. His research has led to the development of innovative vaccines against infectious diseases and new treatments for autoimmunity. The second recipient of the Harrington Prize is Dr. James Crowe from Vanderbilt University. Dr. Crowe received his MD from the University of North Carolina at Chapel Hill, where he also completed his pediatric residency. Following his clinical training, Dr. Crowe received 5 years of postdoctoral training in the Laboratory of Infectious Diseases at the NIH. He completed infectious diseases fellowship training at Vanderbilt and has run an independent laboratory at Vanderbilt since 1996. He's currently professor of pediatrics and pathology, microbiology and immunology, and the Ann Scott Carell Chair at Vanderbilt University Medical Center. His laboratory has a broad portfolio of work in the area of viral immunology and antibody sciences, and his research has focused on adaptive immune responses to various viral pathogens, in particular influenza virus, HIV, dengue, RSV, and COVID. His group, along with Dr. Nuzenswag's group, pioneered the groundbreaking work that enabled the use of human monoclonal antibodies to treat COVID-19. Together, these two investigators advanced the discovery of human monoclonal antibodies for many of the most pathogenic viruses that cause human disease. Please join me in congratulating Dr. Crowe and Nuzenswag for receiving the Harrington Prize.
Hi, I'm James Crowe at Vanderbilt University Medical Center, and so excited to join you today. And my team and I are very honored by the prize and grateful to share with you our work on human antibodies for infectious diseases. Now, antibodies have been around for over 100 years. And in fact, some of the early work we did that most taught us about the human immune system was studying 100-year-old people whose first exposure to influenza was during the 19 epidemic when a parent died in the home and so on. So we took blood cells from 100-year-old people and we were able to find circulating memory B cells that still recognized the 1918 influenza and inhibited. This is a three-dimensional structure done in collaboration with Ian Wilson's lab at Scripps. And in the center, you see a space-filling model of the influenza surface protein, the hemagglutinin, and decorating that are 3 copies of the human FAB, which is an antibody fragment we recovered from the B cells of a 1918 virus survivor. When we looked on the right at the footprint or the epitope of the antibody, we could see all the residues, and we understood the specificity. Interestingly, you'd think, why would a 100-year-old person still have a B cell floating around looking for 1918 virus to come back? That virus has not circulated since at least the 1950s. So for 60 years waiting for a virus to come back. But in fact, 1918 influenza did recur in part during the 2009 H1N1 pandemic, in which the 1918 virus and a bird virus and a human virus and a pig virus, they all sort of mixed in probably pigs because they'd all been sitting there or through exposures. So the 2009 pandemic virus was actually in part 1918. So it turned out the immune system of those 100-year-old people was actually quite wise to spend the metabolic cost to maintain that memory through lifetime. Once we learned this, we knew your body is basically a library of everything you've ever seen. So now we use this type of B cell technology to identify antibodies for any infection for whom we can find a survivor. So this is a recent experiment we did. It's actually unpublished still, in which we took a survivor of Ebola virus infection during the 2014 West African outbreak, and we isolated a large amount of blood using leukapheresis, and then separated out millions of memory B cells, and we were able to use Ebola reagents to sort out 15,000 new monoclonal antibody gene sets from one specimen from one person. And we do that by taking blood, we do a gradient isolation, then we do a paramagnetic bead selection using B cells. So now we have only the B cells, letting the other immune cells go. Then we use flow cytometry by labeling the B cells with an Ebola reagent. We can physically sort the Ebola virus specific cells. And then finally, using single cell RNA-seq technologies, we sequence the antibody genes from those cells. And once we have those sequences, we can do bioinformatic analysis of the diversity, the complexity, the size of the Ebola virus specific repertoire. And so here, Elaine Chen in our lab did bioinformatic analysis of what are called clusters or even lineages. So one antibody somatically mutates and becomes a little family or lineage of antibodies. And so each color indicates different features of these antibodies. So we looked at the binding to 1, 2, or 3 different Ebola viruses, the killing of those or neutralization against each of those viruses. And there's so much information we can gain. This is sort of like the next generation of the Human Genome Project. We're calling it the Human Immunome Project, to look at all of the antigen-specific antibody genes in the human repertoire, and it's vast. So here we obtained 15,000 monoclonal antibodies from one person, and the bioinformatics indicates that we did not even really get to the bottom of this person's Ebola repertoire. Now, one of the interesting things, since we're doing this type of sequencing and other groups are as well, now there are a lot of antibodies that have been published for Ebola, including the antibody sequences. So this is a Circos plot in which we've taken our West African survivor donor, who is shown here on the right, the first 12 to 4 o'clock on the Circos plot. And on the other aspects of this chart are indicate sequences from adenovirus vaccine recipients or VSV vaccinees or survivors who were treated at the Emory Treatment Center. You'll remember there were US expats who were brought back from Africa who were infected. And looking at the repertoire of all these people, we can say, do any of these individuals share antibody genes with other individuals? And so the lines connecting through the center show shared antibody gene usage between one individual and another. And it was really exciting to see this because antibodies are shared between even a relatively small group of individuals. And that bodes well for things like vaccines, because that means you and I might make exactly the same antibody in response to a provocation like a vaccine. So this sort of explains why certain vaccines or certain vaccine antigens work well in a lot of people and others not so well. So we're starting to get a deep view of the specificity and the genetics of the human immune system. Now, how did we get 15,000 antigen-specific cells for Ebola glycoprotein? I'm just going to show you a recent experience we had with COVID. So when COVID entered the United States, we activated our B-cell discovery platform team, and we made antibodies for SARS-CoV-2 using some of the first individuals in North America who had suffered and recovered from the infection. So on the left is a cartoon indicating we have memory B cells and they have the B cell receptor or antibody that they will later secrete on their surface. We labeled that with spike protein or receptor binding protein from that spike protein that we made recombinantly. We sorted those cells out physically, and then we activated them, simulating what might happen in a lymph node or a germinal center. So those cells started making the antibody that they encoded. And we put that on a microfluidic device, in this case, Berkeley Lights Beacon device. We also use 10x genomics. There's different ways to do single cell activity, but this is a view, it's basically a micrograph looking down onto the chip on the Berkeley Lights instrument. It's an actual experiment. So I'm going to show you a movie that was taken over 30 minutes. And of course, we don't have 30 minutes, so I'm going to show it to you in a few seconds. But each of these vertical indications is what's called a pen. It's like a well, and there is one B cell in each of these. And because they're stimulated, they make antibody. And so if I'll start the movie, and you'll see over 20, 30 minutes, antibody comes out of each compartment holding one cell, and the antibody decorates antigen coated beads out in the main channel. So it's like a volcano spewing out antibody, and then we can detect it by fluorescence. So underneath each one of those volcano eruptions at the very bottom is a SARS-CoV-2 specific cell. And we use the instrument light tweezers. So just light is enough energy to grab the cell, move it out in the channel, and capture it. And now we have the genes for an antibody from one cell. So this kind of recapitulates that. On the left, we isolate human peripheral blood mononuclear cells. Then we do some kind of single cell selection. And then we sequence the genes from those single cells. We work with Twist Bioscience, which is a company that synthesizes DNA at very large scale, so they can do thousands of gene synthesis on a chip, sort of like a desktop printer for DNA. And then we take those DNAs in a plasmid and we put them into mammalian cells, and we can have a protein made within 2 days. So this is a super rapid workflow, and it's super high throughput. And by doing that, we were able to make thousands of antibodies very quickly. And this is just the workflow of how we characterize those. The top left, we developed a real-time virus neutralization assay in which we did rough screening at dilutions of 1 to 20, 1 to 100, 1 to 500, and 2500. And we could see cells growing and staying healthy, or if the curve goes down, the cells are dying. And so we could estimate the potency of antiviral activity, which is called neutralization. We also, on the bottom left, were doing combinations of antibodies. We knew we would use combinations to resist escape of the RNA virus, but we were looking for synergy or cooperativity, because ultimately we developed a combination that had that property, which is very beneficial. We work with Jesse Bloom's lab, top right, to do deep mutational scanning of the antigen and look for loss of binding in a yeast system in which every amino acid that could possibly occur in the receptor binding domain was introduced. We saw very little escape for some of these antibodies. And finally, we did EM and crystallography, and we could see our lead antibodies binding at the same time on the RBD. So we were able, in March of 2020, in 25 days, to go from a blood sample in a FedEx box to transferring the sequences to our pharma partner, which was ultimately AstraZeneca. And we did that drug discovery program in 25 days, and that became the drug that's listed at the bottom, 2 monoclonal antibody names. They're also known as Evusheld in the combination, and they're now used under EUA. They've been given to millions of people for prophylaxis against SARS-CoV-2. So that's just snapshots of how we do it. We are doing this for 100 virus targets in a program we call AHEAD 100. And these are just examples of structures of antibodies we've made in recent years for various viruses. And we've gotten leads for about 40 viruses of the 100 targets. So in summary, using single B cell studies these days, we can do really deep repertoire mining, tens of thousands of antibodies. We can do this extremely fast, like SARS-CoV-2, 25 days. And also we've launched a program called AHEAD 100, which is why don't we prepare for epidemics ahead of time, lay down and stockpile these types of antibodies so that we'll have them ready on day one rather than months later. And in summary, we think antibodies are increasingly the future of preventing and treating infectious diseases. Certainly we need vaccines. We will have vaccine development going on, but this is a tipping point in history where we see antibodies becoming a really important player in this space. And we're hoping to continue to contribute to it. And thank you so much to Harrington for recognizing this body of work. And just to remind you, good science is team science. This is our team. Not everyone worked on SARS-CoV-2. We've got so many viruses going, there are different expertise. Many of the collaborators are listed at the left, commercial parties we worked with, funding, and even our local celebrity, Dolly Parton, contributed to our work. And thank you so much for listening.
All right, I want to start by saying it's a great honor to receive this prize. It's really something really very special, and I'm very appreciative of the Harrington Institute, not just for the prize, but also for all the support, the work that we did on the SARS coronavirus. So I want to spend just a few minutes explaining how we got to where we are and what we've done to facilitate the development of antibody therapeutics. So I'm a B-cell biologist, and I became interested some time ago in the problem of why there was no vaccine and no cure for HIV. Vaccines, of course, are mediated by antibody responses, and being a B-cell biologist, this was really a mystery that I wanted to learn more about. And what we started with was knowing that some few individuals that are infected with HIV develop serum antibody responses that are able to neutralize the virus and, in fact, prevent infection. And so what we wanted to learn was, what are these individuals doing? How are they making their immune responses so that we could potentially learn about how to reproduce what they do, or at least understand their antibodies? So to do that, what we did was to collaborate with clinicians who had access to these individuals, and we recruited them to Rockefeller University in order to study their immune responses. And to do that, we developed a new kind of technology, a technology which would allow us to capture the cells that were making the antibodies that were in the serum of these people, the memory cells. And to do that, what we did was to take the surface protein on HIV, the target of the antibodies, and use it to identify the B lymphocytes in the circulation of these people, these very few cells seen here, isolate the cells as single cells, and then use molecular biology to reproduce their antibodies in vitro, give us access and test them to see what they were. What happened as a result of that and the adoption of this method by many laboratories throughout the United States was that we learned that people like this can make antibodies that are very, very potent. So this is a neutralization, and this is the percent of strains. So HIV is very variable, but these antibodies are neutralizing nearly all strains and at very, very low concentrations. And this is something that had not been known until this technology was applied. Now, having these very, very potent antibodies available led us to ask some questions. Can they be used for protection? Can they be used for post-exposure prophylaxis, for chronic therapy, or even eradication of this disease? And so we wanted to do that because antibodies have properties which are a little bit different from drugs, which are highly available and inexpensive, but don't cure the disease. And the property that antibodies have that drugs don't have is the ability to engage the immune system, to clear viruses, to kill cells that are infected with viruses, or to activate antigen presenting cells to make them better at making CD8 T cell killer responses. We started this as clinical trials, or in fact, initially in animal studies, with two different antibodies. We picked them because they are non-overlapping epitopes. They see two non-overlapping epitopes, which would make them resistant to escape mutations for one or the other. We did a series of experiments in mice initially, and then macaques, and then in humans. And in humans, we've infused these two antibodies into over 300 different individuals. What we've learned from those studies is that antibodies cloned directly from humans by these methods are both safe and effective for prevention and therapy in humans. And these antibodies and others like them have been licensed by major pharmaceutical companies that are developing them for therapies for HIV. More importantly, however, this established a paradigm. It established the paradigm that antibodies can be obtained directly from humans that are producing these antibodies that we can see in serum, that they can be cloned rapidly from B lymphocytes that you obtain from blood by tagging the protein of interest to make it identify cells that bind to that protein and then sort them as single cells. And because they were already natural human products, it would be very highly likely that they would be safe if given to other human beings, which is something that you can't know if you make a hybridoma antibody from mice, or if you make a drug from first principles. So this is a tremendous advantage. And it's fast. It's much faster than the typical way that we used to make antibodies for human therapies. So what that would involve is immunization, tissue culture fusion to make hybridomas, selection. Then these are mouse antibodies, so they would have to be humanized. And that's a process which takes quite some time and is not always successful. And finally, because these are made in mice and not in humans, we would have to test them to see if they are in fact reactive to human tissues. And then cell line production and manufacturing. The difference here is that the screening of the sera is something that's very rapid. Flow cytometry, extremely rapid. PCR cloning, again, very rapid. And expression and selection is something that just takes a couple of weeks. And because these have been pre-screened through humans, again, it's very highly likely that they would be safe. So when SARS came around, we were prepared to try to reproduce what we had done for HIV, and others adapting these methods also proceeded down the same line of thinking, which is to use the domain of the spike protein, the RBD, that interacts with the cellular receptor for the virus ACE2 to use as a bait, to capture antibodies that might interfere with this. And that enabled, those technologies enable the cloning of many different antibodies to SARS, their classification as different entities, and more importantly, an understanding of how these antibodies might select for the variants that we see coming up in nature. It also allowed us to learn a great deal about how humans respond to these vaccines and how their responses broaden over time, explaining why these vaccines are in fact so good. Moreover, this technology enabled many companies and ourselves included to develop antibodies that could be given to humans as rapid therapies before drugs became available, saved thousands of lives. So What we've done is developed a method and a paradigm for rapidly producing antibodies that can be used clinically. They can be used for passive protection. They can be used for chronic therapy. They can potentially be used for eradication of diseases. And they are being deployed and developed for SARS-CoV-2, of course, we know that, and are in late stages of development for HIV-1, hepatitis, Zika, tick-borne encephalitis virus, and even malaria. Thank you very much for your attention.